Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 161st seminar, and I call it University of Pennsylvania Day. It is already May 1st. I cannot believe it. I hope April was the coolest month, as a poet T.S. Eliot described, meaning from now on, things are getting better because we hit the lowest point and have a path to move up. I'm attending the annual Engineering Biology Research Consortium, or EBRC, meeting at University of Washington in Seattle to discuss the future of engineering biology or synthetic biology. It is great to meet many of my colleagues, friends, and trainees here. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Cesar de la Fuente. He is a presidential associate professor at University of Pennsylvania, or UPenn. I think I got to know him a few years ago, and when I looked at his publication record, I thought he should be already a full professor. He got numerous awards. I'm not gonna, list those things. Today, he is still very young, but a true pioneer in his field, especially the antibiotics related field. Cesar, thank you so much for your great contribution to the scientific community, and the virtual podium is all yours now, and thank you again.
Great. Thank you so much for that kind introduction and for having me really in this space. It's really a pleasure. Let me share my screen and we can get going with the presentation. All right. So what I thought I would do today is tell you a little bit of the work that we've been doing over the years, trying to accelerate antibiotic discovery using AI and experiments, experimental validation. I think antimicrobial resistance is the greatest existential threat to humanity. These are bacterial infections that are really difficult to treat or even untreatable. They're currently associated with 5 million deaths per year in the world. And the projection, if we don't come up with new antibiotics, is that by 2050, that number is going to double to 10 million deaths per year in the world. As you can see in this graph, that is projected to surpass every other major cause of death in our society, including even cancer. So it's really a tremendous problem. So throughout my whole career, I've devoted a lot of time and energy in trying to come up with out-of-the-box approaches to counter drug-resistant infections. Additionally, the problem is that it takes a long time to discover even preclinical candidates. It can take up to 6 years, which, to put into context, is more than the time that it typically takes to complete a PhD program. So it takes a long time, and it's really expensive to develop new antibiotics. And so a number of years ago, we came up with this, I think, fundamental idea of, could we use AI to accelerate antibiotic discovery? And this was breaking away from the traditional paradigm that we typically, or science has typically used for identifying new molecules, which is a really a physical process where you actually, scientists go around nature and you take soil samples or water samples, and then you try to purify potentially active compounds that might be present in all of that complex organic matter. As you can imagine, this process is really painstaking, and it relies on trial and error experimentation, and oftentimes does not actually yield any promising candidates. So instead of doing that, we decided to take advantage of the decades worth of biological data that we have at our disposal in the form of genomes, metagenomes, and proteomes that have been sequenced over the years. And we decided to think of biology essentially as a bunch of code, a bunch of letters, right? If you're thinking of DNA, you have nucleotide letters. If you're thinking of proteins or peptides, you have amino acid letters. And if you think of biology, if you conceptualize it as a bunch of code, then we can develop algorithms to mine all of that vast amount of code to try to identify new potential antibiotic molecules or bioactive molecules that might be encoded within that complexity. So the analogy is a little bit like looking with a magnifying glass, zooming into biology, into that complex code, and identifying potential molecules that might be, the next therapy tomorrow or the next solution to some of the problems facing humanity today. So I'm going to tell you just briefly how we did this. We started by mining the human proteome for the very first time as a source of antibiotics. The human proteome is all the proteins encoded in our genome. So we're essentially talking about our entire genetic code and genetic information. Using an algorithm, we explored all of this code and we identified thousands of new small proteins called peptides that have antimicrobial properties, many of which were previously unrecognized to have any sort of antibiotic properties or any role in the immune system encoded in our proteome and in our genome. So this was really exciting. In fact, we've shown that some of these bioactive peptides that we've discovered, they're actually they play physiological roles. They're produced at different levels in our body, as we can see here. And we think that we call these encrypted peptides, and we think that um, they represent a hidden layer of immunity. So we think there's all these peptide molecules encoded in our body and in our genome and encoded within our proteome that are playing a role in defending the host, in this case ourselves, from invading organisms. And so we're continuing to test that hypothesis. This work really sparked a lot of new ideas in my lab, one of which was the fact that we're finding all these compounds in the human body. It probably means that they're not just there, not just confined to the human genome and the human proteome. So we decided to explore the idea that maybe a lot of these small peptides that we're finding, maybe they're conserved throughout evolution, and maybe we may be able to find them throughout the tree of life. And since we had done this initial work with the human body, we decided to see if we could identify similar compounds in our closest ancestors, which are Neanderthals and Denisovans. And in the process of thinking about this, we came up with a framework for molecular discovery that we call molecular de-extinction, whose ultimate goal is to identify molecules, namely small peptides and proteins that are produced or have been produced throughout evolution, to then be able to synthesize them in the laboratory using chemistry, and then learn how mutations or changes that occurred over time in those particular molecules influence their biological function. And their biological function, can be antibiotic properties, functions in the immune system. It could be functions in the nervous system, anti-cancer properties, you name it. And so this is really a conceptual framework for thinking and studying molecules throughout time. So going back to ancient humans, to be able to identify antibiotics in archaic hominids, we developed a machine learning pipeline that we call PanCleave, that what it does at a very high level, it takes all the proteins from, let's say, Neanderthals, it chews them up in little fragments through computational proteolysis, and then we ran a number of filters, including human expert filters and machine learning filters, to predict whether those fragments might be a promising new antibiotic. This framework enabled us to identify the first therapeutic molecules in ancient biology and in extinct organisms. And we can see some examples here of molecules identified in Denisovan proteins and in Neanderthal proteins. But of course, this is only theoretical work. So then we had to actually validate this in the lab experimentally. And in my lab, we're lucky because we have an AI machine learning team, but we also have a chemistry team, and we have a synthetic biology / microbiology team. So we can actually experimentally validate everything that we discover on the computer. So we actually synthesize a lot of these small peptides using chemical methods, and we validated them in the lab. And we saw that some of them were able to kill bacteria that we grow in test tubes, and also have anti-infective properties in different preclinical mouse models of infection. So this really was exciting. It demonstrated that we could apply this idea of molecular de-extinction in a practical way to identify bioactive molecules. So it gave us the courage to try to actually ask a more fundamental question or ambitious question, which was, why not just sample all of ancient biology for antibiotic discovery? But this it involved actually coming up with a more powerful AI model because it was a scaling problem. We're going from here a couple of organisms, right? Modern humans, Neanderthals, and Denisovans. But if we want to explore all of ancient biology, we're talking about hundreds of organisms. So it's a lot more data to sort through. So we developed Apex, a new AI model from scratch in my lab, and we trained it also from scratch using experimental data sets that we've been collecting in my lab for a while. This is the architecture. I think the really amazing thing about Apex is that you give it an amino acid sequence, and it directly predicts the antibiotic function from that sequence. So it's a sequence to function prediction model. It doesn't really take into account structure. So it's arguably really hard, right? Really hard to directly predict sequence from, a function from sequence. And Apex really, it opened a window into the past. It enabled us to sample through all of ancient biology, including the Holocene and the Pleistocene. Here I'm showing representative organisms where we identified some of the top hits that Apex discovered. It includes penguins that were extinct in the 50s, ancient zebras, magnolia trees that disappeared throughout evolution. It includes ancient elephants, woolly mammoths, giant sloths that existed, tens of thousands of years ago to over a million of years ago, and many other creatures from the past. So really, it enabled us to explore ancient biology for new bioactive molecules. We then synthesized a lot of these things that we found, and we tested them in preclinical mouse models of infection. And I'm showing two different mouse models here. This is a skin abscess infection model, and on the bottom we have a deep thigh infection model, which is typically the one that the FDA in the US, the Food and Drug Administration, wants to see for promising candidates. And I won't go into all this data here for lack of time, but the top candidates that we found came from the woolly mammoth, a molecule that we've named Mammuthocin 2, and also from the ancient sea cow, giant sloth, and the extinct giant elk. We had to come up with new names for all these compounds that we're identifying, and some of them, like the ones named here, were really promising in this preclinical mouse model. So we're really excited about the potential of this work. In terms of next steps, like I mentioned, we think of biology as a bunch of code that can be searched with AI. And so we want to mine all of it, all of biology. One of the dreams that we had was to actually mine representatives across the tree of life. And the tree of life is composed of three main branches: eukaryotes, which we focus a lot of our attention initially in the form of the modern and ancient humans. But then we also wanted to see if we could discover antibiotics in the microbial world of bacteria, and also in the domain of life of archaea. And I don't have time to go over all these results, but we have now sampled the global microbiome and human microbiomes. And also we have a preprint on digitally mining all the archaea of the world for new antibiotics. And all of this work has led to the discovery of many new candidates. So sort of to summarize, using traditional methods, it can take many years to come up with new preclinical candidates. But today, in my lab, using AI and experiments, in a few hours, we can discover hundreds of thousands of candidates. And if we really push our resources, we can go from discovery to synthesis to testing within 1 or 2 weeks. So it's really it's an amazing playground for a scientist like myself. I've dreamt of this moment for a long time, where we could really speed up the process of antibiotic discovery. So it's really fun to do this. And in terms of the impact of Apex and our other AI models, it has essentially multiplied the speed of discovery by a factor of several million. It has enabled us to, by mining all of biology, including living and ancient biology, discover millions of new candidates. And it has saved many years of human research time, right? This work, this acceleration. And people have estimated that this use of AI for antibiotics has saved the equivalent to thousands of PhD students working for 6 years each. So it's just amazing to see what we can do nowadays with machine learning and having the right data sets to train those AI models. And with that, I'd like to thank lab members, past and present. It's really a privilege to work with them and learn from them every day. Our amazing collaborators from all around the world, our funders that allow us to do the work that we do. And here I'll just leave you with links to our website, email, and social media in case anybody wants to get in touch. And again, thank you so much for this opportunity. It's really a pleasure, and I look forward to the Q&A later.
Fantastic. This is a pretty amazing. And now people talking about AI, and then now I really see, one great example what, AI or computation could do for biology. This is amazing. And then in that way, I hope, there are, a lot of people saved, in the future, because of this technology. Thank you so much.
Thank you so much. Yeah, that's my hope that we can apply AI to biology and medicine and hopefully benefit humanity in a very positive way.
Fantastic. Yeah, absolutely. Yeah, amazing. Thank you. All right. Now the main speaker of today with a longer introduction, Hajar, or we call Nika Estiri, is an accomplished researcher in the field of molecular medicine and synthetic biology. She holds a PhD in molecular medicine from Tehran University of Medical Sciences, where she graduated first in her class. Her doctoral research focused on innovative gene therapy approach for epilepsy. And currently, she is a postdoctoral fellow at the Craig Venter Institute in California, working in Dr. John Glass' synthetic biology and bioenergy group. Here, she lead a project on engineering the skin microbiome for innovative disease management. Previously, she conducted postdoctoral research at the Latvian Institute of Organic Synthesis, where she worked on enzyme engineering and biocatalysis. Her research interests span synthetic biology applications, gene therapy, bioengineering approaches to drug delivery and immunotherapy, and protein engineering. With a track record of academic excellence and cutting-edge research experience, she brings, of course, the valuable insight into the intersection of molecular medicine and synthetic biology. Nika, thanks so much for your time today, especially with a lot of other things you need to do, including family care. So thank you so much for your time today. And please take it away and all yours now. Thank you.
Thank you. Thank you, Taisa, for the kind introduction. I want permission to share my screen. Can you see my screen now?
Yes. It's coming up for the screen. I see right now. Can you click that button?
Sorry?
The screen share, the slide mode. The bottom, yes. Right now I see your left PowerPoint as well as the main one, but you could click the slide mode. Yes, that's that one.
Is that correct now?
It's kind of slow. It's not coming up. I don't know why. Do you see the slide show mode right now?
Okay, I share again if
Yeah, that probably better.
is that okay now?
I don't think so. Last time it worked when it tested. I mean, the one way you can do is make the side figure smaller and then main one bigger. In that way, people could see. But you could try. Now there you go. Okay.
It's okay now?
Yes, yes. Now it's
Okay,
okay now.
perfect. Hello, everyone. My name is Nika Estiri. I'm a postdoc here at JCVI Venture Institute, La Jolla, California. And today I'm excited to share one of the main projects that I worked for 1.5 years here at JCVI. And I'm happy to be part of this SynBYSS community, this international, young, talent and motivated scientific community that try to make the world a better, a healthier, safer, and hopefully a happier place to live through biology and synthetic biology. Since joining JCVI, I had privilege to work with John Glass as my mentor. And today, my aim of my presentation is to want to have a flavor of thinking of Staphylococcus epidermidis, this skin microbiome as a platform for therapeutic protein delivery. Yes, I'm talking about Staphylococcus epidermidis, this gram-positive bacteria that lives deeply in your skin, friendly without triggering any immunologic reaction. Then I joined to work on this project. This project actually defined and it was narrow to engineering this skin microbiome as a surrogate for type 1 diabetes, as a better management for the type 1 diabetes. But I would like you to have this picture of this potential in your mind. So by looking this title, maybe 3 words will highlight in your mind: engineering, skin microbiome, diabetes. Maybe quite is odd, but I want to explain the motivation behind this project and why this project was started. Why engineering a skin microbiome? It's because the scientists discover Staphylococcus epidermidis that exists under a few millimeters under the skin, the place that before was believed that there is nothing or a sterile place. But in this discovery actually was done by our collaborator, Dr. Richard Gallo at UCSD, who is well-known dermatologist, and his work and his team, their discovery in 2013 led to extend our vision to the dermis and the discovery in bacteria. So this bacteria is deep enough in the skin that it will be in contact with the capillary vessel and can sense the blood. So this was the discovery. And the next step, why engineering this bacteria? Because it's not a model organism, it's a Staphylococcus epidermidis. It has a thick layer of the peptidoglycan, is a gram-positive. Who wants to engineer this non-model organism? JCVI, as you know, as a pioneer in the microbiome research, start to work on engineering of this bacteria. And in the JCVI, John Glass, that is my mentor, he is with having 2 decades of experience and pioneering in synthetic biology with Yu Suzuki, who is an associate professor also at JCVI, determined to propose this proposal and start to work on this intractable bacteria to transfer to a tractable and genetically manipulatable organism. So why engineering a skin microbiome for diabetes, not other disease? The thing is that unfortunately diabetes is ranked 3 of causing death in United States, and this demand and this number of increasingly of the patient demand a new innovative and better management for the diabetes, and this attract 1.6 million fund for JCVI and overshadow other disease and other metabolic disorders. Maybe they are actually easier target, but the diabetes it is a big target, but of course it's a hard target. Then I will explain what is hard to explore this potential. JCVI, as a leader of this project, collaborate with other institute and university, including UCSD, Stanford University, Bioengineering Department, and also Indiana University. And thanks to the foundation of this project, Larry Hillblum Foundation is a foundation in California that funds the research project that is focused on diabetes and neurodegenerative disease. So this project actually proposed as an alternative for traditional treatment that exists for the diabetes. As you know, the treatment of diabetes and the recombinant insulin is expensive, is invasive, although there is a smart device like an insulin pump, still is a mechanical device and needs continuous monitoring of the glucose for the patient. So this, on the other hand, engineering of the skin microbiome has this potential to be an alternative and cheaper treatment, non-invasive. Biological control versus mechanical control can be done by this approach. It can reduce the discomfort of the patient, and the most importantly is that is invisible to immune system. So diabetes as a increasing of the blood glucose in the blood for the long term, and insulin that is used for the treatment of the diabetes. Actually, insulin works as a key for the cells, because in the presence of the insulin, the presence of this key, the glucose will enter the cell, and in the absence of this key, this essential key, the glucose will remain in the body, circulate throughout the body, and damage from eyes, heart, and kidney, and so on. So as you know, the insulin actually produced from the beta cell, and this beta cell is responsible to producing insulin. In the presence of the insulin and binding insulin to its receptor, it's allowed to glucose enter the cell. And this project wants to be a surrogate for the beta cell to this staph epi works as a mimic or a surrogate for the beta cell. In the diabetic patient, especially type 1 diabetes, that include 10% of the patient, this beta cell actually is attacked by the immune system. So there is no beta cell or functional beta cell, or there is no enough insulin to produce. So the patient have this struggle and increasing of the glucose in their blood. On the other hand, type 2 diabetes that include 90% of the patient, although the insulin is produced, again, the level of the insulin that is produced is not enough, and the cells are not responsive to this glucose. So again, the patient needs to inject the insulin. I was looking for, because I working on I was interested in the cell therapy, and advanced therapy is something is new for can be done for diabetes. So maybe someone asked, there is for example, advanced therapy in reprogramming to producing induced pluripotent stem cell. Is there a way can come up with a solution for the diabetes? So it is fascinating. For example, you can isolate cell from the patient, from the skin or fibroblasts. You can treat the cell with a reprogramming factor, and after a few weeks, you can have a pluripotent stem cell that potentially has this ability to differentiate to different cells, including beta cell. And this beta cell can be transplant to the patient. But the problem is that this beta cell, especially in the type 1 diabetes, that is produced and reprogrammed and is fresh, it can produce insulin. But the problem is that this cell, again, will recognize by the immune system, and they are not safe from the attack by the autoimmune system. So the patient needs to, during their whole life, use these immunosuppressive drugs, that if the patient is so young or is in elderly age, it can be problematic. So what is our roadmap, or what are our blueprint for therapeutic skin delivery? Now I want to explain what is these steps, and I will bring you briefly through this step that we have done so far. The first step is that we need to identify an insulin variant that can be produced by the bacteria, because insulin, as you know, has a post-translational modification. So it needs to be produced with bacteria, so it needs to be functionally and biologically active. The next step we need to engineer a staph epidermidis. We need to manipulate and make it tractable and genetically change the bacteria to can introduce our DNA or our construct that's including the insulin. We need to confirm that this engineered bacteria, it can penetrate, still this ability is intact. It can penetrate under the skin and it can reach to the dermis to be deep enough to can be in contact with the capillary vessel. The next step is we need to demonstrate that this functional insulin can not only can express in bacteria, it can secrete to the outside the bacteria inside the for example, if it is in vitro in the media or it is in vivo inside the blood. The next step is we need to first test the activity of this single chain insulin or insulin analog that we have to see how it works in vitro and then hopefully in vivo. And the next step is we need to develop a smart glucose responsive biosensor because we want to use this biological control versus mechanical control. This bacteria will need to be able to sense the glucose. The next thing, and maybe most important thing, is to establish a biosafety and biocontainment measurement to be sure that we can control this engineered bacteria. So the whole idea is that we take the Staphylococcus epidermidis from one of the non-pathogenic strain, and we manipulate engineer this bacteria in such a way, and we introduce a DNA responsive element inside the bacteria that this bacteria can sense the level of the glucose. If the level of the glucose is, for example, in physiologically relevant in the high concentration, it can, after sensing the glucose, it will be fast and will be adaptive to this response to can secrete insulin analog into blood. And after secretion, this insulin analog should be efficient enough to can adjust the level of the glucose to the normal level. So this is the whole idea of no more needle that was proposed for this project. So I want to go through the steps through this blueprint that we have for this to explore the potential of this bacteria for better management of type 1 diabetes. First of all, we need to identify an insulin variant. As I said, insulin has a post-translational modification. How we can overcome this challenge? This part of work has done with collaboration with Dr. Michael Weiss at Indiana University, who is a structural biologist, biochemist, and expert in designing and synthesizing several analog of the insulin, including single-chain insulin 57. As you know, just shortly, you know that the insulin is composed of two chains, A chain and B chain. A chain is composed of 21 amino acid and B chain that is composed of 30 amino acid. And these two chains is connected together with the 3-disulfide bond. That two of them is between two chain and one of them is inside the A chain. So there is 3 disulfide bond. And as you know, the environment inside the bacteria is a reducing environment. So it's hard to have a disulfide bond. This is another challenge that we want to find a solution for that. Dr. Michael Weiss find a solution for this problem. And as you know, the insulin produced as a proinsulin inside the cells, in the beta cells, that has a C-peptide that during the post-translational modification, this C-peptide will remove and A and B change will join together with 3-disulfide bond, and then they found the 3D structure and the proper folding. He replaced this C-peptide with the linker of 6 amino acid. So create a single chain amino acid that can produce in one chain from N terminus to C terminus, and this adding 6 amino acid increase the number of amino acid to 57, and the name of this analog actually back to the number of amino acid in this analog. So The idea is that we take this sequence of this single chain insulin analog. Now we have this potential insulin analog that can be produced in bacteria, because this chain A and chain B are close enough together that after secretion outside of the inside the bacteria, inside, for example, in the media or in the blood, because now it's a non-reducing environment, this disulfide bond can be formed, and there's a potential that the insulin can find the functional and proper folding. So we took this sequence and this insulin analog, and already Dr. Weiss showed that this insulin analog, single chain insulin 57 and other variant, is quite equivalent to the native insulin. He did NMR and other studies and even checked the biological activity and binding to the receptor of the insulin, for example, and even did in vivo study by injecting inside the mouse. So for now we have our candidate for the insulin. So the next step is how we want to engineer a staph epi, because as I said, it's a non-model organism. The thing that we done, and it was successful, we try to manipulate the gene, and we knock out one of the gene that is responsible for the degrading of the enzyme, degrading of the DNA. Because when you transform this bacteria with your plasmid or with your construct, the problem is that it has a type 4 restriction enzyme that cuts and degrades the DNA. By knocking out this gene that is MCRR, that is stand for methylated cytosine recognition and restriction enzyme, by knocking out this enzyme, we were able to increase the efficiency of the transformation to 1,000-fold, and it was make our life easier to can introduce the next step, our construct that is insulin. So the next step was that has done is that we confirmed that our engineered bacteria that is now, for example, knockout for one of the restriction enzyme, we introduce first reporter gene, different reporter genes, including superfolder GFP, and also we work with the MK2, that is red fluorescent protein. We introduce our construct inside the staph epi. Then we paint this bacteria on the skin of mice, and 72 hours after painting on the skin of mice, we did section, and we were able to detect our bacteria that is under the skin in the dermis, and it was successful that this bacteria still keeps these natural properties. So the next step is that demonstrate the secretion of this insulin analog, because now we have this construct we introduce in the bacteria. How we can be sure that our insulin is produced in the bacteria and it can be secrete the outside of the bacteria. To detect the secretion of the single chain insulin, I design a vector, and I create this plasmid that actually has a secretion signal. And after secretion signal has a single chain insulin variant, that here it shows another variant of the single chain insulin, that is single chain insulin B. Along with that, I add the nanoluciferase sequence. That is, nanoluciferase is very sensitive, and it's very good to detect this enzyme. By detecting the activity of this enzyme in the supernatant, I was able to measure the activity of the nanoluciferase, and I show that this actually this single chain insulin is secreted outside of the cell. And because I purify also this protein because I add also his tag construct at the C terminus of this sequence, so I was able to purify my single chain insulin from the supernatant, and I do the downstream in vitro assays. So for in vitro assay and for checking the activity of this single chain insulin, I used the HepG2 cells. This is the hepatocellular or human liver cancer cell line. This cell line has an insulin receptor. So the single chain insulin, if it binds to its receptor, it can trigger a signaling that I can detect as by Western blot. So the thing that I did, actually I used insulin as my positive control, and I used the single chain insulin 57 as my analog. And after exposure of 15 minutes to these cells with the insulin, I did the extraction of the whole protein, and I did Western blot to actually detect the downstream of this activation of this processing of after activation of the receptor. Just a brief background. When insulin binds to its receptor, is trigger a cascade of the phosphorylation inside the cell that ultimately lead to the entering the glucose inside the cell. One of these protein that has its play role in this cascade is AKT, is a protein kinase B. So I try to do Western for this protein, and by confirming of this protein, I will be able to check the activity of the insulin analog. So I did Western blot. So I had two groups, control and versus treatment. The control group was the group that doesn't have any treatment with the single chain insulin analog or insulin. And the treatment was exposure to different concentration of the single chain insulin 57. And I did SDS page and then Western blot with the antibody against AKT and phosphorylated AKT. That for phosphorylated AKT, I used the position serine 473. So as you see in this figure, the AKT, the level of the expression between the control and the treatment is almost the same, but with exposure to increasing the concentration of the single chain insulin and this analog, the level of the phosphorylation compared to the control is increasing. That it shows that this our single chain insulin that is produced, expressed and secreted by single chain insulin by staph epi is able to activate this signaling pathway. So it shows that it's functionally, biologically active. So the next step is to check the activity of this insulin analog in vivo to be sure that to see how it works in vivo. But before to recheck this extensively check on the model of the mice, we need to develop a smart glucose responsive biosensor, because we want our staph epi work as a biosensor to firstly, if there is a physiologically relevant high level of the glucose, to be responsive and coordinate to the level of the glucose. So to do that, we here I need to explain this part that I said diabetes is a big target, but it's a hard target. Why? The reason is that we need the precise level of the insulin, because too level of the insulin, it will be lead to the hypoglycemia and it's life-threatening. On the other hand, too little insulin is not effective, and it keeps remain the pathological effect of the high level of the glucose. So the challenging part is here that we need to find this precise DNA responsive element that we can use as a biosensor. This work is something that is under ongoing process that has working with Stanford University bioengineering group, Dr. Drew Endy and Kaisha Benjamin. Actually, Kaisha defended her thesis, and her thesis was dedicated to identify a DNA response element or transcriptional promoter in reaction to elevated of the glucose. So just a brief background, as you know, transcriptional promoter is part of our DNA that determine when, where, and how much gene needs to express. So in our case, it's this glucose sensitive promoter. How it works, the first step is needs to be highly sensitive to can detect the level of the glucose, and in coordinate with the level of the glucose, it will be able to activate, and upon activation of this promoter, it can lead to the expression, transcription, and translation of the downstream gene that in our case, for example, insulin analog can be produced and secrete outside the cell. So to do that, to find this glucose sensitive promoter, The thing that Kaisha did at Stanford, she cultured staph epi in the presence of the different concentration of the glucose in a biologically relevant concentration. Then she extracted the RNA and did the sequencing library. And after sequencing, she tried to analyze the transcriptomic data. And this figure that you see here is a figure that she published last year. In this article, she showed that in the presence of the spike level of the 17 millimolar glucose in the cell culture, she discovered that 43 genes will be upregulated and 42 genes downregulated. This kind of genes that are significantly show upregulation or downregulation, it can be a candidate for our glucose responsive element. So this is something that we need to screen, we need to find out, but for now we have some candidates for these DNA elements. The other thing that we need, maybe the most important, is we need to establish a biosafety and biocontainment protocol to prevent the spread of the engineered bacteria outside the lab or in the healthy people, not in patient. So what I mean by biocontainment, imagine if we have a patient that he received this engineered bacteria, this Staphylococcus epidermidis in the patient can, after contact with the healthy people, it can transfer to the healthy people. But the biocontainment, it allows us to manage this and control this bacteria in such a way that it can survive on the patient, but it cannot survive on the healthy people. So this is that we need to do, establish this biosafety protocol. For this, we create a strain of the staph epidermidis that is auxotrophy. It means that this bacteria, it doesn't grow. Otherwise, we need to add some additive in the cell culture. And we deleted and we knock out gene thymidylate synthetase A. Is a gene that it's in the production of the thymine, that is one of the building block of the DNA. So by knocking out this gene, the bacteria, if we culture in vitro, they will able just only survive or grow on the plate that has a thymidine. In the absence of the thymidine, the bacteria will not grow, so it cannot escape from the lab. For in vivo, imagine if we have a cream, that this cream is including a bacteria that has this construct of the insulin that is able to produce insulin. It can be used as a cream on the surface of the patient's skin, but in this cream it also has this additive thymidine to support the growth and survive of this bacteria. So the patient needs to frequently use this additive and this cream to keep the bacteria that is engineered keep in his microbiome on his skin. So this is we are not there yet, but this is the whole story and whole the roadmap that we designed for our experiment. But some of this, I didn't show some of the data because it's unpublished research. I want to publish, and then people that are interested, they can go deeper inside. But like any other translational project, this project has a challenge, including scientific, safety, and efficacy challenges, ethical and regulatory challenges, and practical and logistical issues. So as a scientist, now I'm focusing on the scientific, safety, and efficacy challenges. One of the important things is that about the gene regulation, how we can identify optimal glucose responsive element to be relevant to the physiological level of the glucose, and will be fast and will be responsive to can precise control the level of the glucose. The other thing is biocontainment. We need to have a robust biocontainment strategy. Maybe we need to add more kill switches along with the auxotrophic strain. The other thing is that therapeutic efficacy, how we can achieve clinically relevant level of the insulin that is proper, has a proper folding, and is biologically active. So at the end of this seminar, maybe you are curious and you want to know this technology, if this technology has this potential to replace the traditional treatment that is exist for the diabetes, like a injection of the frequent injection of the insulin or insulin pump or monitoring of the glucose frequently. The thing is, the answer is not clear yet. We are not there yet, but I believe that the research and the tools that has developed during this project has this potential to revolutionize other metabolic disorders, the other disease that they don't need the precise control of the level of the insulin. So it can be helpful for other disease, for the skin inflammation. Imagine we have a staph epi that can produce anti-inflammatory cytokine, and then by painting on the skin, for example, by producing IL-10, it can reduce this discomfortment and this inflammation. The other thing, for example, as a cancer, as a vaccine for the skin cancer, like a melanoma, that actually this work was done in Stanford by Dr. Michael Fishback and his group, that they show that it has a potential to use as a vaccine for the melanoma. So I want to just have this picture in your mind, that this engineering of this skin microbiome has a potential to be explore more maybe in the future soon. So by this, I think I'm done, and I want to say thank you for your listening and your patience. And I am happy to have answer any question that you may have.
Fantastic. This is amazing. I also started, skin microbiome project with my collaborator. Actually, we do use another bacterium called C. acnes. So we potentially collaborate in the future. Let me ask Cesar, he has any question or comment before I entertain one question from audience.
No, I thought it was fantastic. Thank you, Nika, for the presentation. Yeah, I really think this could be really promising in the future, right? To incorporate some of these engineered microbes onto creams and things like that. And the idea of engineering microbial communities is something that my own lab has worked on. And imagine if you could, in a predictable way, engineer communities and be able to shape them right over time in beneficial ways. I think that's obviously it's an amazing goal to have, right? Because we're surrounded by these microbes, and we have so many of them living on our skin that if we're able to control them in some predictable way, I think that would be really cool. So yeah, congratulations.
Thank you. And I can imagine
on your research.
I can imagine that we produce one of these this peptide that you see as an antimicrobial. We produce in this staph epi, then paint, use as a vaccine. I don't know. I mean, it can be an alternative for not, for example, the delivery, because oral delivery for protein is hard. But for example, painting on the skin will be much easier, I think.
That is
absolutely
exactly what I want to say. I mean, for the delivery of your peptide. Using this bacteria might be one potential collaboration.
Yeah, let me know if you're interested. Would love to chat more.
Sure, absolutely. So I have one question from audience. Bruno asked question. Sorry if this is a basic question. There's no basic question. Everything is great question. But thinking about real world applications, would this bacterium be applied to the skin every day? I believe, if so, wouldn't regular washing or, taking shower potentially remove it? That's the question.
Because this bacteria is naturally exists. We do the same, we wash our hands every day, we go, shower or everything, but this bacteria is something, it go under the knees of the skin. If a few millimeter and it go to dermis, it's not only in the we paint, but maybe you need to, for example, a few hours, you should stay to, I don't know how much in the human, because it's hard to translate from mouse to human, but if you do the clinical trial, for example, in human to see how much it takes time to go to reach to that place. Then we have a window. Maybe in this window you need to be care, but after that, I think it should be the life will be normal.
Yep. So basically, the skin skin microbes do colonize relatively well compared to the gut microbiome microbes. So that might be a beneficial thing, but at the same time, we need to also start to worry about just GMO. I mean, as you mentioned, the biocontainment and then longer colonization is not necessarily good thing because we don't know what kind of thing happened with GMO.
Yeah.
So that's great answer. I have one question. It's also related to translation in the future. So I do understand, glucose sensor concept. We also have the glucose sensor in my lab, so I'm happy to, collaborate with you regarding glucose sensor development. But the question I have is, basically bacteria do exist in the skin, but not not, in the blood, stream. Actually, the bloodstream is not good. I mean, bacteria in the bloodstream is not good. But the question I have is, how are you gonna correlate between blood glucose level? That's the most important thing with the whatever level in the skin. This is not easy, question to answer.
Yeah. This is, I mean, need to, I said this development of this biosensor, we have some candidates, but we need to test how it works. And especially in I know, in the human, I think it's really quite different from the mouse, to translate this information. But it's something that's really is need to work on.
Yeah, so at the end of the day, I mean, whatever concentration this bacteria detecting in terms of glucose level, we need to
Yeah,
know that how much in the blood, level, and then we're
exactly.
gonna do a lot of experiment to potentially AI kind of answer some of the question, basic, question. I mean, answer that one, hopefully,
Yeah,
but that some, experiment need to be done
sure.
in the future. Let me check whether we have question. Oh, let me see. No, I do not see more question from audience. So in that case, I mean, I just going to close and then we probably just start to chat. Okay, so thank you all for joining and staying today. We'll meet again on May 22nd, Thursday, the same time, the same Zoom link. Please note, we have 3 weeks of break. I call it, spring break or summer break, whatever. We'll have Dr. Alessandra Ferrari, a research program officer at European Research Council or ERC, and Dr. Amelie Scope at Fulbright. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who could speak and show your handsome and pretty faces if you wish. Thanks. I stop recording. 1 second.